So good morning, everyone. I'm really pleased and honored to be the first speaker of this day. And I think my talk is mostly probably an overview, not an overview of what we know. And I really like the beginning of the slides because there's so much to be learned, too much understanding, and really expand our horizon. And I think the microbiome is a very beautiful example of that because we are realizing its importance. We're realizing it's going to be something that we're going to have to assess and treat and so on. But very important is that it's really part of the whole. It's not just one individual thing. So here's just my disclosure slides. But I think what is important to understand that a microbiome, we need to probably change our understanding of GI physiology. And we change GI physiology because we want to change our understanding of whole physiology of the organism. Because as you can see, there's a complex ecosystem in the GI tract. In the past, we thought bacteria are mostly pathogens or we don't care about them. Now we're realizing it's a really important part of the whole system. And it's important to understand that it's not isolated. The GI tract is not just there's bacteria, there's the host, it's one ecosystem and all of them interact together with each other. So when we look at the physiology of the GI tract, we have here the intestinal epithelial layer, we have an important mucus layer here in yellow. We have a very complex and diverse bacterial ecosystem. And if everything is okay, we have a really well-regulated and balanced immune system. When you look at this is a slide that actually one of my PhD students recently made, and I think I really love it because it really shows you that we need to not just look at the epithelium here, but this is the same thing. Here is the intestinal contents. Trillions of bacteria. It's an ecosystem that is really present in us. It's actually very difficult to manipulate. Then here you have this very important mucus layer here in black. It's probably one of the most underrated safety feature in the intestinal tract. And then we have the epithelium here. But important to understand is it's not one or the other. I think a lot of microbiome science goes, let's look at the microbiome, but often forget that the microbiome is really one part and both interact with each other. So that means, for example, if we have changes in the microbiome here, and again, when you look at the sheer numbers, it's actually not that easy to change this ecosystem. It's like a jungle out there, it's like 100 trillions of bacteria. But if there's changes here with antibiotic-induced dispersal or something like that, if everything else is here okay, it's probably not such a big deal. The body has a huge buffer capacity. But if the system breaks down, if you have inflammation here, architectural changes like we see often in chronic enteropathy, then ultimately that's going to ultimately change the gut microbiome, and then suddenly we have this combination of different factors. So it's important to understand in microbiome research, in GI research, and actually I think in any other organ system, that we have to consider this. And also understanding why does the microbiome change. Second thing is the microbiome is really a metabolic organ. I think we're realizing more and more. It's important in immunity, but it's more important probably the immunity via metabolites. So when you think about it, that's why nutrition is so important. Every single thing that enters the GI tract, it can be macronutrients, medications, drugs, somehow it's going to be metabolized by bacteria because they are hungry. They love to eat, they love to feed, and ultimately all those little metabolites they're going to produce, they're going to be absorbed along the entire GI tract, and ultimately we're realizing more and more that this becomes a centerpiece and ultimately all those associations with different conditions and so on. Is it cause-effect? I think it's a combination of both. It's going to be very difficult to separate it out, which makes sense because it's one ecosystem. But in the end, I think it's important that we need to probably realizing that. And I think more and more data really comes out that harming the microbiome long-term really becomes a risk factor for many diseases. In human medicine, where they can really look at major studies, we can look at 10,000s, 100,000s of individuals over time, there is clear associations that change the gut microbiome, mostly to antibiotic, because that's probably the strongest factor we can change it, or are associated with many of those chronic diseases. But again, realizing it's also a small risk factor. It doesn't necessarily mean we should never use antibiotics. It's just realizing there is a combination of factors, and we need to see the organism as a whole, not isolate it out. So my talk today is going to be a little bit of a kind of a hopefully overview. What do we know? And my thought was always that it's an ecosystem, and when you look at your own ecosystem in your own backyards, there's a lot of small things like weeds and so on, but even ecosystem, it's probably the most important are the major plants, the major trees and so on. And I think it's the same thing probably in the microbiome in the guts. Some of the major metabolites, like short-chain fatty acids, bile acids, and tryptophan metabolisms seem to really be account probably for the majority of changes, if it makes sense. And we're going to focus on those. And then ultimately also, microbiome dysbiosis does never come by itself. it's mostly always secondary to something. The ecosystem goes off if something changed. And we know now the most common causes are antibiotic usage, some dietary deficiency to some degree, and ultimately, I think, intestinal inflammation, chronic GI disease, and so on. So I'm going to talk about this a little bit. So again, there's a lot of complicated metabolites, a lot of bacteria out there. But really, when you start reducing literature to major pathways, we really come out to that some of them are more important than the others. I mean, it's maybe too early to say, but I think some of them are always associated with clear associations and so on. We're also realizing that some bacteria are always keep coming up in literature. So we're realizing those bacteria seem to be the important ones. So for example, one of the most famous ones are the short-chain fatty acids, which comes from dietary sources, carbohydrates, fibers. Bacteria can ferment them into so-called. But also important is it's this balance. And I think when we always focus on something is good, we also need to realize something too much good can sometimes be bad too. And there's always this balance between not enough of those metabolites and too much. And I think short-chain fatty acids are in good concentrations are really very helpful, then anti-inflammatory, regulate motility. But ultimately, if you have different ratios of short-chain fatty acids in dysbiosis, if the ratio of acetate propionate is different, they can actually activate virulence factors from enteropathogens and so on. Another important pathway that keeps coming up most recently is the tryptophan pathway. Again, various bacteria can do it. Tryptophan gets metabolized to indole metabolites. And again, in right amounts, they're actually quite beneficial. They're anti-inflammatory, maintain insulin barrier function, but if there are too many of them, they can also be harmful. We have other, now I was realizing that some amino acids can be converted to bacteria, ultimately, like P-cresol, for example, again, ultimately can act as rheumatic toxins and progress to kidney disease. So ultimately, a lot of different metabolites. So let's focus on a few of those. So what do we know about the GI tract in microbiome in dogs and cats? And again, one thing that is also, for me, interesting and really highlight is that, yes, it's a very complex ecosystem. There's a lot of bacteria out there. But if we really start reducing literature and also data into ones, we discover there's actually so-called core microbiome. And what that means is there are some bacterial species that are consistently present in healthy animals. And there's a lot of other ones that are there, but again, they may be important, but again, focusing on some of the major ones can help us really cutting through the noise. So I'll give you an example. So this is a study we just recently did. We looked at shotgun sequencing. That means we can really identify bacteria on a species level. And yes, we identified almost 1,000 bacterial species across 12 healthy dogs. But when you look carefully, those in green are positive, present, those in red are absent. You can really see that really there's actually only a few bacterial species that are really consistently present in every single healthy animal. Okay? So 25 of them were all identified in all healthy dogs. And if you start looking which actually bacterial groups are always present, we're realizing it's kind of always the same, like Faecalibacterium, Clostridium hydrononis, Blautia, and so on. And we're starting realizing what their functions are. And we're going to talk about all of them. But we know that Clostridium hydrononis is a very important bile acid converter, so this pathway is very crucial. Blautia, Faecalibacterium are bacteria that really produce short-chain fatty acids and other peptides and so on. So again, it becomes more and more clear that we're trying to understand what are the major pathways, the major players, and they can really move us forward in the field. So why is short-chain fatty acid so important? Most bacteria that are in the colon have some capacity to produce short-chain fatty acid. I mentioned Faecalibacterium. This is a bacterium that really, and slowly, also we need to memorize some of those bacterial groups as beneficial. That's important. It's not just random bacteria. Those are important health bacteria promoters. But Faecalibacterium has been in human medicine already. Many different studies associated with health, with inflammation and so on. It's an anti-inflammatory bacterium that produces some peptides that can reduce inflammation, but it also can produce short-chain fatty acids. And short-chain fatty acids really activate in the lumen, activate different receptor epithelial level, and ultimately can dampen and downregulate inflammation. There's very clear studies associated with decrease in short-chain fatty acid producing pathways and inflammation. So this pathway is an important one that we want to ultimately bring up to dampen inflammation. The problem with that is always that we need to also understand, again, coming back to the first slides, it's not one or the other, okay? It's always a combination. There must be a risk factor. Otherwise, if it would be just a simple thing that we change something, I think we'll all be dead by now. It is this combination. There must be a predisposing factor. The body is over millions of years, we have evolved in a very protective system. So meaning there must be dysbiosis. We know there are nice studies in animal models. If you take the dysbiotic microbiome from a patient with IBD and put this into a mouse model that is susceptible to developing IBD, it's actually going to develop inflammation. So we know a bad microbiome can definitely, stimulate things. But again, there must be a cofactor. There must be some underlying damage, some other factors that ultimately drives its importance. But again, bringing it back to maintaining health, it makes also sense not to be dysbiotic, if that makes sense. We shouldn't really, only because we cannot always induce it, it doesn't mean just being dysbiotic is a good thing long term, because it's a risk factor. And what makes it actually even more interesting is that there's realizing that many of those receptors, we thought it's just in the GI tract, right? It's just GI immunity and so on. But suddenly there's data that shows that actually short-chain fatty acid receptors are also located on kidneys, So again, it suddenly makes this confirmation that what's happening in the GI tract truly has effects outside the GI tract. So the GI tract really becomes an important key organ system with potential risk factors for other organ systems. We know now that bacteria, some bacterial species can prevent allergies, for example. Again, those are studies done in germ-free animals. But if you're in germ-free animals, for example, they have no bacterial species, you feed them with some of those allergens, they can become allergic. If you feed afterwards specific bacteria, there are an association that some bacteria are more protective versus other ones. And short-chain fatty acids are also very important in prevention of, for example, controlling enteropathogens. And I think that's from a clinical perspective an important one, because very often we got trained to always look at the pathogen, right? If there's a Clostridium overgrowth, that must be the pathogen, so let's just give antibiotics and so on. Not realizing that very often it's this dysbiotic environment created in things that actually allows overgrowth pathogens. But they may not be so important, The goal would be rather more bring the microbiome back, and that's going to eliminate by itself all those potential pathogens and so on. So the other important pathway that really comes more and more, and I think it's really a fascinating one, is actually bile acids. So we always think about bile acids, liver disease, digestion and so on. But bile acids are actually becoming really one of the most important signaling molecules across many different organ systems. And why is that? It's actually a beautiful also example, this symbiotic relationship between the host and microbiota. Because this conversion of bile acids that come from the liver as primary bile acids, and a small percentage of them reach afterwards the colon, and as far as we know at the moment, only bacteria can really convert those into secondary bile acids. So it's a beautiful example that bacteria are part of our physiology, okay? Because that's all it can do. And this conversion of this primary secondary bile acids is important because the proper ratio of those bile acids, and mostly actually the secondary bile acids, those microbial converted bile acids, have a lot of key functions. They work with different receptors, like FXR receptors, that signal is back to the liver. There's this DGI-5 receptor in other organ system. When they get activated, they mostly reduce inflammatory signaling and so on. There's a nice data in humans and animal models that actually also bile acids can potentially regulate insulin glucose secretion via some of those receptors in the pancreas. And again, coming back to this, the microbiome really also reduces enteropathogens, because if you have the proper secondary concentrations, they're going to reduce Clostridium difficile. If you have dysbiosis or something, suddenly you have an activation of them. So again, it becomes really an important pathway. And in dogs and cats, I'm going to mention this a little bit later, really very fascinating, only Clostridium hieranonis is this beneficial bacterium that is strongly associated with this conversion. So it's one bacterium that becomes like a very important key species. And just to show you those bile acids, again, it's a crosstalk. It's not just the bile acids by itself, but many of the health receptors, even vitamin D receptor to some degree, gets activated by those bile acids, and ultimately, as you can see, tons of different health functions across different organ systems. So again, it's not just one or the other. There's a lot of overlap between different pathways and so on. And it makes sense. That's not just one single pathway. Otherwise, again, if you eliminated one pathway, we'll be in deep trouble. So there's a lot of different crosstalk between different receptors, different metabolites that come from the host / from microbiota. And Paula Garretta, she's now a pathologist, but when she did her PhD, when we started looking at those bile acid receptors, as you can see, this is the TGR5 bile acid receptor. So this one is mostly activated by secondary bile acids, and with immunohistochemistry, she stained it in a small and large intestine of healthy dogs. And you can see how ubiquitous it is. The expression of those transporter receptors are really pretty much everywhere. They are in, for example, in ganglions, endothelial cells. We see them in the pancreas and so on, macrophages. So again, this pathway definitely seems to be a very important one also in dogs and cats. So this conversion of primary to secondary bile acids. There's also very good data in animals, but also in humans. So where you see that this conversion, the bile acid metabolism is really complicated. There's probably at the moment, I think 50 different bile acids, subtypes identified. But the primary principle is that you have primary bile acid from the liver, you have conversion to secondary by the microbiome, and then there's even a little deeper that even the secondary bile acids can be converted by some bacteria into subcategories and so on. So it gets a little bit more complicated, but generally the principle is the same, that we need this conversion to some degree by bacteria, and ultimately we have dampening of inflammation, suppressing on inflammatory pathways and so on. However, as I said before, not everything is good. They're great, it's the balance. There's definitely also, if you have too good of something, it can go in a negative direction. There are definitely some associations with, for example, in other organ systems, with lung cancer and so on, where an overexpressed receptors can actually be activated and ultimately may be carcinogenic. We also know that also from human medicine, that's why bile acids already got so confused, because the past, if you have a very high fat, high protein diet, you have too many bile acids, ultimately they're irritating and they kind of cause also, colon cancer associations. So it's not like, it's the balance, and it's very, very important. But then also, for example, you start seeing those, associations with other organ systems. I was just at a conference a few days ago where there's a colleague of ours from Missouri, and she does actually looking at the lung microbiome, lung disease, and she says they see a lot of those associations between gut disease and lung disease, We always think they're isolated, but think about it, sometimes you have this bile aspiration, it's completely possible, like in this, and ultimately, in this case, you can actually trigger the negative effects and so on. So we need to understand all of those are overlapping, it's not just one or the other. So important to understand also what actually causes dysbiosis, what is the most common reasons why we have those changes of those important bacterial taxa. As I mentioned before, over time, there was a reason we identified which groups are usually in disease, decrease in disease or dysbiosis, which groups go up in disease, because those are the core species. And so over time, we were able to identify some of those core species. We understand a little bit more of their function. We also know we have now reference intervals for them, and so that really helps us to also much better understand shifts in the microbiome. It's not just a little bit, is it there? We can define much more what is a more normal versus abnormal gut microbiome. So we can measure that, and we can also actually measure with the so-called dysbiosis index, which puts all those data together in one single number, the extent of dysbiosis. So the higher this dysbiosis is, the more the microbiome shifts away from normal. The less diverse it is, we see very good correlation between diversity and increased dysbiosis index. And it's I think once you have an increased dysbiosis index, many of those animals are going to stay abnormal for a long time, because for me, and I'll come back to this afterwards, it's almost like the system is already broken. Once we see detected dysbiosis, there's probably also really severe GI disease, because again, the microbiome is always a reflection of some underlying disease process. That is important to understand. And often the first marker, because it will be exposed to the entire GI tract, is like an ecosystem. When the trees are dying, something is dying. It's not by itself. There must be something, and in most cases, it's the soil, the quality of the soil, the separation, and so on. And interesting is we also can see slowly, because once you have a targeted assay, once it's a reproducible assay, we can slowly see it's not a random thing. We can differentiate to some degrees acute from chronic GI disease, and meaning that in acute episode of diarrhea, the index, we have shifts a little bit in the microbiome, a little bit increase in Clostridium perfringens, in coli, but the entire system is not yet completely out of balance. For example, versus in chronic GI disease, and again, I'm going to come back to this a little bit. In chronic GI disease, there's a long-standing process, meaning there's a villus atrophy, there's short inflammation, and that actually really destroys the ecosystem, and it's difficult for it to come back and so on. But acute, and so you can see that in most chronic GI disease cases, we have really much more increased, less diverse microbiome, increased dysbiosis. We very often have decreased Clostridium hydrononis, this bile acid converter. Whereas in acute diarrhea, yes, we have diarrhea, but diarrhea doesn't mean dysbiosis. Diarrhea simply means an acute self-limiting thing. In most cases, it's a minor shift in microbiome. It normalizes very quickly. And I'll give you an example. So we have now 3 studies done in Europe where we can really see in acute self-limiting disease, we see an increase in perfringens. So there's a little mild-grade dysbiosis, but not enough to really shift the normal gut microbiome way into abnormal ranges and so on and so on. So it's important that we discovering a little bit, it's not just this black bucket, diarrhea is diarrhea, dysbiosis, it is definitely some underlying differences and so on. So what is the major driver of microbial changes that ultimately also going to cause metabolic changes and potentially could be risk factors for the future. And I think we all know by now it's actually antibiotic usage. And that's really something that until a few years ago, we always thought, well, you have dysbiosis, you give antibiotics, we're going to fix the dysbiosis. And it's interestingly because antibiotics, when you look at all the markers out there, yes, it works clinically in some animals, but every other marker of health actually gets worse. That's really a fascinating topic and so on. But ultimately what you can see that here, those are studies that we summarized together. So this is actually in the red, metronidazole induces a huge shift in gut microbiome. Tylosin here in black, as you can see, takes some animals come back really quickly after 1 or 2 weeks, but in some animals, it takes really over weeks to really come back into normal. It doesn't only change the dysbiosis index. You can see the Clostridium rhamnosus, important group, is really affected by broad-spectrum antibiotics. And again, many animals don't really ever come back to normal in this important metabolic pathway. We also see some differences between antibiotics. In dogs, interestingly, amoxicillin clavulanic acids doesn't really induce a big change, surprisingly, but in cats it does. So there's definitely also differences in animal species. Cats, I can tell you, are very, very sensitive to antibiotics. And in cats, the microbiome stays much, much longer abnormal, even compared to dogs. So that's important also to understand. But I think important to understand is that some of those taxa that we know are beneficial, we know they provide metabolites of good ones, they are severely affected by the use of antibiotics. And see here's in cats. This is also a new recent study we published in the Journal of Filament Medicine. We have a dysbiosis for cats now, and you can see also many cats with chronic GI disease have dysbiosis, but antibiotics, especially amoxiclavulanic acid, as you can see, changes a lot of those bacteria. E. coli goes up, Clostridium rhamnosus goes down, and so on. So cats really are very sensitive to those microbial changes, and they're long-lasting, much longer-lasting than in dogs. So that's one. So antibiotics, obviously it is. Again, I want to come back to this principle. It's not so easy. We should not say we don't use antibiotics if we need to. Okay, it's just a risk factor. But I think it's just realizing there is a potential, and it makes a cost-benefit analysis in which conditions do we truly need and so on. And I think we can all agree in GI disease, in most cases, most cases really do not need antibiotics at least. There's other therapy options and so on. But the other really important pathway we showed you before, in acute diarrhea, we have this really minor shift, very transient and so on. But in chronic GI disease, we have a shift, a major shift, and it's a long-lasting shift. And the reason for that is, again, in a healthy gut, it's normal. You have this mucus layer here, you have a closed epithelium, you have normal microbiota. In chronic GI disease, and again, that's really a huge overlapping conundrum, But generally, most cases, even if they're mild, when you biopsy them, they're going to have some architectural changes, villus atrophy and so on. And ultimately what happens there is that you're going to have, I'm going to show you a few slides afterwards, but the overall thing is you have inflammation, we have to some degree malabsorption because many of those transporters, receptors, going to be decreased. And all of this together really drives dysbiosis and so on. And that's why this is very long lasting, because the gut structure itself does not heal that quickly, okay? Even does it ever normalize ever? So that's why often, even animals go into remission, the dysbiosis pattern stays. And again, it's just a marker of the underlying GI disease. And ultimately, it's my theory at least, often even with diet, we're managing the disease, We're reducing substrate for bacteria, but ultimately it's not necessarily curing the disease, okay? Because the gut structure, once it's this way, it's almost impossible to bring back to normal, at least until now. And I give a nice example. This is a healthy dog. Again, here in red are bacteria in the lumen. This black layer here is the mucus, here's the epithelium. So you can see in a healthy dog, you should really have this separation of bacteria from the mucus layer. And this is one dog. This is an extreme, so that's not like every dog's going to look like that, but you can see beautifully that here's the epithelium in blue, and then here there's no separation, there's no mucus layer, and here in red are all the bacteria that are severely attached to the epithelium and so on. And you can probably appreciate that even with antibiotics, we're probably never going to get rid of this biofilm, number one. And that's why we have this high relapse rate, because we don't really able to cure the disease. We can suppress the clinical signs, but probably it's never going to normalize and so on. So I think it's an important way of understanding the pathophysiology and also prognosis and so on. And here is another beautiful example, again from Paula Garretta. And so when she did her PhD, we also looked at bile acid transporters. And so on the left side, you see the villus of a healthy dog in the ileum. And we can see how beautiful long it is. And then here in brown, that's actually the brush border. So in brown, we stained the transporters for bile acids, which should be absorbed in the small intestine. And for me, it's actually a beautiful example also how superficial this layer is, okay? And it's not just the bile acid transporters, they're transporters for like amino acids, for many other compounds. And you can appreciate it in GI disease, in a chronic enteropathy case, we have the architectural change. It's not only the inflammation here, but it's the architecture, the function of the GI tract is reduced, and you can also see this reduction in those transporters, okay? And again, when you look at this on the right, how likely is it that these villas were ever going to be beautiful long and all those transporter functions are going to come back? So I think one thing you also need to realize, we have this long, low-grade malabsorption in the small intestine, and that ultimately, if you have malabsorption, what's going to happen? Bacteria are going to drive on that. So that's another form of dysbiosis. So dysbiosis is not always inflammation, not always change. These architectural changes, they are persistent, they really drive the microbial shifts, and then it can actually contribute to clinical signs. It's not one or the other, it's a combination factor, but it's definitely important to understand. So coming back to Hydrononis, because it's actually fascinating that this bacterium is such a crucial species. Okay? And I mentioned before, it's this bacterium that is very sensitive to antibiotics. It's this bacterium that really decreases in this long-lasting GI disease cases, okay? And it's a bacterium that's fascinating because we have reference intervals now, and you can see that all over the world, when you measure by qPCR, they're all in this reference interval, okay, most of them. Just a very small percentage of healthy dogs have decreased Clostridium hydrononis, but when you measure afterwards bile acids in those are the animals who do not have secondary bile acids. That means if this bacterium is decreased, it is abnormal because, again, at least to my best understanding, there's no reason not to have bile acid conversion, okay? There's no alternative for that. So we can really associate this also as being abnormal. But again, most dogs have a very, very narrow reference interval in this area. And this is the conversion. We have now several studies. I think it's a very, very strong correlation. It's fascinating that this one bacterium you can predict almost from the qPCR data if bile acid is going to be conversed and so on. It's really a health beneficial bacterium. And this is a study that actually was done by Johnny Lee from Purina, and they looked at sequencing data, bile acid metabolism in dogs with heart disease. And in this study it was very nice because actually you can see that this dysbiosis by itself, if it's high, it clearly separates the microbiome based on sequencing from low and so on. But actually more important, interesting to me is when you look at Clostridium hydrononis, so low here in blue are dogs that have decreased Clostridium hydrononis. Yellow, they have normal Clostridium hydrononis, and you can actually see the separation almost. So just the presence of this bacterium, the presence of the bile acid pathway really allows you to almost give you a good separation of the microbiome. It's such an important bacterium. And that's why also when you look at the dysbiosis index, most dogs who don't have hydronosis are going to have a dysbiosis. So that explains this importance of this one single bacterium, can explain this pathway, and ultimately bile acid pathway. Okay, that's the one important one. And that's important to understand from a perspective of immunity, other organ system, but also enteropathogens, because again, when you think about human medicine, Clostridium difficile is a very important pathogen. And it has been very, very clearly associated with the secondary bile acids, okay? And there are two theories. One theory is that secondary bile acids kill the spores, prevent the germination of Clostridium difficile. But also new data has shown that there's a consumption, there is a competition for substrate. So for example, if you have bile acid producing bacteria, if they are in a certain amount, they can actually compete for some of the amino acids and actually take away the substrate Clostridium difficile. If you reduce them, and again, antibiotics or GI disease are the most common reason they get reduced, then suddenly Clostridium difficile can overgrow, and ultimately that's why it can cause the disease in humans. In dogs and cats, it's a little bit different, and the reason for that is in dogs and cats, for some reason, we do not see the association between Clostridium difficile and GI disease. But we see the association between dysbiosis and the presence of those organisms. And so indirectly, we have to be aware of that, and I'll come back to this in 1 second, is that if it's present in dogs and cats, it's actually more a mark of dysbiosis and so on. It's not necessarily something I should treat. I'm going to come back to this one. But important is that we understand that also the microbiome develops over time. And again, the reason when you're a puppy, well, not you're a puppy, but when we are little, babies or puppies, it's ultimately what happens is that we don't have yet the functional GI tract to maintain the anaerobic bacterial normal microbiota. So when we are born, in the beginning, we take off bacterial species from the environment, and mostly it's E. coli. But here in puppies, as you can see, in healthy puppies, you can see they have E. coli over the reference interval, but 7 to 9 weeks, 10 to 16 weeks, they come into this reference interval, what we see for adult dogs. That's why also Faecalibacterium, this is a beneficial anaerobic species, is not yet present. And you can see in this 3 to 4 months, it comes into this normal range and so on. The reason for that, again, is over time, the GI tract develops pH, digestive enzymes and everything, and that allows the microbiome to develop and so on. And that's why we see the dysbiosis index, it's in this case physiologically abnormal. That's a part of development. So because they don't have yet the proper microbiome, but somewhere in this with 4 to 6 months, they really come into the reference interval that we see in adult dogs. So the microbiome develops, okay? We can see those changes. But again, coming back to why the microbiome is so important is what we saw in puppies, for example, is in the very first few weeks of age, a lot of Clostridium difficile, okay? But no clinical signs whatsoever, you know? Most of those dogs were also toxin negative. But again, we still have so many textbooks who say there's a pathogen, and so suddenly imagine some puppy has diarrhea. You would immediately say, oh, it's a pathogen, maybe we should treat them, and so on. Yeah? But interesting was that you can see 7 to 9 weeks, suddenly you see this sudden drop of it. It just disappears, almost becomes negative, without any really treatment. And why is that? Because as you remember, the normal microbiome is the one that develops at this time. And as you can see, beautiful Clostridium hydrononis is not present here. It starts showing at 7 to 9 weeks. What does it do? It converts bile acid. You can see also beautifully how the bile acid suddenly increases secondary bile acids, and we know that secondary bile acids really control some of those organisms and so on. So again, showing that the microbiome is really, some of those groups and some of those pathways are very well associated with changes in those areas. And we see this also in dogs now with chronic GI disease. Okay? We see in our data that roughly 35% of dogs that have chronic GI disease are positive for Clostridium difficile. But when we went back and have an association with dysbiosis, roughly 90% of those dogs had increased dysbiosis index. Okay? So 90% of dogs who were positive Clostridium difficile had an increased dysbiosis index and didn't have Clostridium hydrononis, so they have bile acids. Okay? And we also looked at the treatment outcome of those dogs, and there was really no association with treatment outcome. Most dogs responded to, like in chronic enteropathy, were food responsive. Okay? So again, in many cases, we know that dysbiosis is a part of the chronic enteropathy complex. We have decreases in some of those beneficial bacteria. We have an overgrowth of enteropathogens, but it's not those were the reason. They just overgrow, and again, based on clinical data, food, dietary trials and so on, are always still the staple of the first one and so on. So again, coming back to this one, we need to understand the microbiome is an important factor in all of this. It's not the only one, but it plays a role. And so ultimately, if you have this damage, and there's damage in the GI tract, inflammation, you have degradation of mucus layer, malabsorptions, you're going to have a dysbiosis. If you have the dysbiosis, suddenly many of those potential enteropathogens we always thought, like E. coli, Salmonella, and so on, they can be activated or they can be present, but that doesn't mean they are the reason of the disease. We need to stop focusing on them so much and really come back and say, how can we go back to an underlying disease process and so on. That's important. There's also nice studies, not from our lab only, but also from Penn State, for example. They showed that, for example, in dogs that are food responsive, the microbiome slowly comes back, but it takes time because this underlying condition is not going to change. But they also showed that over time, microbiome, so the hydronosis comes back, bile acid comes back to some degree. They also showed nicely that actually when you incubate E. coli Clostridium perfringens in vitro with bile acids, the secondary bile acids are also suppressing them and so on. So again, important information, I think, also from a clinical perspective, to not focus so much on pathogens, but understanding what is the reason why we have a dysbiosis, and that's really mostly already a very chronic process and so on. In the very last few slides, I would like to talk about a little bit about tryptophan metabolism, because it's also something that actually is becoming more and more important. And actually, tryptophan catabolism is an important one because it's actually a key amino acid that is really at the center of diets, center of absorption of small intestine, and ultimately being able to be converted by bacteria into so-called indole species and so on. So tryptophan comes from the diets, usually supposed to be absorbed in the small intestine, plays an important role because it can be converted into serotonin, which it's an important metabolite. Can also, to some degree, a small endocannabinoid acid, but also tryptophan that still remains in the lumen of the GI tract can be afterwards converted by bacteria into so-called indole metabolites. And again, only bacteria can do that. And there's a lot of different indole subclassification. And ultimately, when you have inflammation, some of those enzymes can actually shift this pathway in this direction, away from serotonin in this direction, plus the dysbiosis ultimately can reduce the conversion into indole metabolites, okay? And I think from a nutritional perspective, indole always sounds like a bad thing, you know? But I think what we need to understand a little bit, we're talking about a huge metabolites, you know, that is very abundant in the GI tract and has health functions. If you have a little bit too much of it, that might be the negative effects of things, but generally it's actually considered to be to some degree a beneficial pathway. The reason why that is because tryptophan metabolites from indole can really have other direct effects on cells. So they can induce macrophages to be like anti-inflammatory, like interleukin-10. They can prevent conversion into TNF-alpha and so on. But there's also indirect effects. Via some receptors like the aryl hydrocarbon receptor also has garnered a lot of attention recently in human medicine as a potential target to dampen inflammation. So again, this pathway via tryptophan indole metabolites seems to be also an important one and a major one. So one of our grad students, that was in 2016, he did actually a mouse model where he actually induced GI inflammation with NSAIDs. And so the hypothesis was that this pathway, like indole, because it's anti-inflammatory, when you add indole to the treatment, you can actually prevent a little bit the severity of GI, of those changes. And it was very successful. It shows that you're actually adding indole in a certain amounts, it was associated with a less damage to the epithelium. But also interesting is, in the beginning, it was not unclear how much indole can you give, you know? And so the very first experiments were a little bit tough because actually, if you gave too much indole, it ultimately caused more severe damage. So again, coming back to this balance, there's no right or, it's important to understand that. But generally, those metabolites, again, the principle is that a normal gut microbiome that is not dysbiotic, that is there present, converts all of those different, dietary substrates or host substrates into some of those important metabolites. There are data in veterinary medicine. So there's a study from Japan in 2017. They show that tryptophan, again, is decreased in plasma in GI disease. And they also showed actually a correlation with the severity of GI disease. So the plasma concentration of tryptophan correlated pretty well with severity. So the less tryptophan you had, the more severe the clinical activity was in those cats. And ultimately, though, is again, coming back to this balance, is the gut microbiome an important one? The gut microbiome, I think, what I really want to highlight is it's just one part of the whole thing, okay? It is always something that happens, but if it becomes abnormal, then it can contribute. So ultimately, there's also nice data, or more and more data, let's say nice data, especially in animal models. I think there's still a long way to go in humans and veterinary medicine, but the principle makes kind of sense. That's what happens in the guts via inflammation, via components. You're going to have low-grade inflammation, translocation, if the tight junctions go off. You have decreased short-chain fatty acids and things like that. All of this ultimately can also do some damage in distant organ systems. Because again, for example, we know that in the kidney, you have to some degree receptors for short-chain fatty acids. We know if you have low-grade translocation, we're going to have low-grade inflammation that ultimately can affect this epithelium and so on. And we also have, to some degree, conversion of metabolites, like I mentioned before. Tryptophan seems to be very beneficial. But if the conversion is wrong, you can have tryptophan into indoxyl sulfate. And there's data that indoxyl sulfate can progress kidney disease. So again, unfortunately, it's not going to be that easy saying, I'm going to just simply increase this in this disease. It's going to be dependent. But I think understanding the principles is already a very important step. How can we move forward? So ultimately, I think it becomes more that we need to understand in every organ system, is the GI tract normal or abnormal? Okay? Because that can be potentially a contributing factor to many of those diseases. So when we come to the kidney-gut axis, again, unfortunately, not yet much data clinically, and in dogs and cats I'm not even aware of. Only one study from back then from Stacy Summers who showed that progressive kidney disease with the stages, that it seems to also have progression in indoxyl sulfate and P-cresol going up, so there might be a conversion. But in mouse models, they can show that actually increasing some of those metabolites from the guts can potentially be associated with progression. So ultimately, as you can see, I think I'm way on time, the gut becomes really an important pathway in the sense of it is a complex system. It's not just one or the other. We have this underlying structure that's most important. We have no idea really why GI disease happens. It's probably a combination of different factors. It's some damage model, something happens, and then over time, very slowly, the gut develops and ultimately causes dysbiosis shift and can affect many, many different other organ systems. But I think it's important to realize that. It's important to really protect the gut microbiome and try to minimize all those risk factors. It's not going to be a silver bullet, we're going to heal it and so on, but I think minimizing every risk factor or improving every single thing that we understand is probably the future of the world. So with this, I would like to really acknowledge a lot of collaborators, a lot of funding. Without all those data, we would have never been able to assemble all these data sets and also those potential pathways. So with this, I'm ending my presentations, and thank you so much. I think questions are going to be later in the Q&A session.